Welcome viewers to today's session on food microbiology. Under this session, today we will be learning on cultivation of microorganisms. Dear viewers, in this chapter, let us try to understand the various methods for the isolation of microorganisms or microbes. This chapter includes sampling, methods of isolation and cultivation, pure culture techniques, isolation techniques, enumeration of microorganisms, both qualitative and quantitative, isolation of pure culture in media. Let's start with the introduction. Our everyday activities are influenced in some or the other way by microorganisms. Microorganisms may cause and cure infectious diseases. As infections are produced in open wounds by microbes, so also a large number of antibiotics are produced by microbes. In addition, they are used in production of food, production of industrially useful chemicals which are beneficial to humans. The production of food as a result of microbial fermentation predates recorded history. Fermentation was mainly used as a method of preservation, for example, pickle or pickle making, and to increase its taste and flavor of the food. Beneficial bacteria are used as an aid in producing desirable characteristics in food. Some applications of microorganisms are as follows: food products. Under this, alcoholic drinks, dairy products, bread, vinegar, pickled foods, mushrooms, single cell proteins. Products from microorganisms. Under this, we have enzymes, amino acids, antibiotics, citric acid. Mining industries. Under this, we have metal extraction, desulfurization of coal, and under the agriculture, we have microbial pesticides, herbicides, nutrient solubilizing organisms like nitrogen fixation, phosphorus and potash solubilization, etc. Under the environmental bioremediation, wastewater treatment, nitrogen fixation, composting. Let's start with sampling. Sampling requires detailed steps ensuring that the sample procured is a fair sample that accurately reflects the constituents of the bulk material being sampled. The basic principles that must be addressed in sampling are as follows. The sample should represent the food as sold to the consumer. Number 2, each part of a divided sample should be the truly representative of the original. Number 3, the sampling process must not alter the sample in any way that might affect the analysis. Number 4, storage and transportation of the sample must not alter the sample in any significant way, whether through contamination, loss, deterioration or any other means. The nature of the samples that are appropriate will depend on the purpose for which the analysis is undertaken. The quantity will vary according to the product and the type of analysis to be carried out. In addition, samples must be drawn under the provision of regulation that have their own provision. Some of the examples are the poultry meat. Within that, we have water content regulations 1983, SI 1983/1372 number, the milk based drinks Regulations Act 1983, SI 1983/1514, the animals and animal products. Under this, we have examinations for residues and maximum residue limits Regulations 1997, SI 1997/1729. Natural mineral water. Under this, we have spring water and bottled drinking water regulations for the year 1999, SI 1999/1540. Contaminants in food regulations 2003, SI 2003/1478. Sampling regulations as per FSSI 2006. Based on the regulations and given guidance, minimum weight of samples should be taken and packed in relatively small containers. They should be in the sealed containers. At least 6 to 9 items should be taken as sample. Generally, one sample of a specific food is taken and divided into 3 final parts of the sample. On some occasions, multiple samples are collected and preserved separately. A. Administration of sample. This will normally include the use of appropriately designed record forms, sample labels, and laboratory submission sheets, keeping an exact record of all the data. On taking a formal sample for analysis, the sample must be labeled. The label should contain the source, the name of the sampling officer, the name of the authority, the place, date and time of sampling, and an identification number. All the samples should be placed in suitable containers and secured with a tamper-evident seal. Sufficient labels, forms, containers and seals needs to be taken to the point at which the sampling is to be carried out. It is often useful to place the parts of the sample in a second container, such as a plastic bag, which should be sealed in such a way as to ensure that the sample cannot be tampered with without being obvious. In some cases, specialized equipment might be needed to carry out the sampling, and it could include items such as sampling spear, the specific pieces of sampling equipment, temperature monitoring devices, scales and weights. A camera is a useful piece of equipment for recording both the premises and the details of the samples taken when appropriate. The sample should be taken during normal working hours. Sampling visits should therefore be planned as part of the inspection program and timed, having regard to all relevant and available information, which includes hours of operation, hours at which the certain foods may be handled, and any seasonal factors. It is desirable to sample at different times of the day and manufacturing and packing premises to ensure that samples are obtained from beginning to the end of a batch. Transportation of samples. The collected sample must be sent to public analyst or analyst department, particularly where tests are to be made for. It is common practice to transport chilled or frozen samples in cool boxes or refrigerated vans. It is essential to ensure that frozen food stays below 0 degree centigrade and chilled food stays below 5 degree centigrade. Storage of samples. After analysis, the retained final parts may need to be stored for several months prior to the submission to the concerned authorities. It is important that they are appropriately stored. Final parts of food which are perishable should be kept refrigerated or in a frozen state as necessary. Homogenization of the food sample. Food sample must be homogenized for enumeration of microbial contents. The stomacher, a relatively simple device, homogenizes specimens in a specific plastic bag by vigorous pounding. Do 2 paddles, figure 1. The pounding affects the shearing of food specimens and microorganisms are released into the diluents. The other method is using a high speed blending. Methods of isolation and cultivation of pure culture. Let's learn on this aspect. Microorganisms can also be used as indicators. Microorganisms like E. coli that are usually associated with the presence of pathogens are called indicators. Most E. coli do not cause illness, but they are indicators for fecal contamination. Therefore, there is a need for food safety control hazards to the consumer. Microorganisms can also cause foodborne hazard that may cause a food to be unsafe for human consumption. Therefore, food quality controls deterioration of food. The development of microbial isolation techniques makes it possible to use pure culture in food production, isolation of these bacteria and identify them. It is essential to maintain the originality to know organisms. The study of pure cultures opened up the possibility of classifying microorganisms on the basis of the characteristics they display in pure culture. The first step in the cultivation of microorganism is therefore the creation of a pure culture. A key development for the production of pure culture was the ability to grow microorganisms on a solid medium. Koch had noticed that when a nutrient surface such as cut potato was exposed to air, individual microbial colonies grew up, and he inferred from this that these had each arisen from the numerous divisions of single cells. The use of agar was first suggested by Koch's colleague's wife, who had used it as a setting agent in jam making. Agar does not melt until near boiling point, hence an ideal setting agent, resisting both thermal and microbial breakdown. Shortly afterwards, Richard Petrie developed two-part culture dish that Wash named after him as Petrie plates. Petrie plates can be sterilized separately from the medium and provide protection from contamination by means of its lids. This is still a standard equipment today, although the original glass has been largely replaced by pre-sterilized disposable plastics. Let's learn on isolation techniques. One of the first requirements to study specific microorganisms is to separate them from the mixed microbial populations in which they are found in the environment. To achieve this goal, microbiologists use culture media and aseptic transfer techniques. Let's learn about inoculation. It is an introduction of a very small sample of cells called inoculum into a respectable containing nutrient or culture medium. The aseptic or the sterile technique is a technique designed to keep the working environment as free of contaminants as possible. This is achieved first by sterilizing all equipment and media that will be in contact with the microorganisms. This includes minimizing the air movement on the working area. Usually, the work is done within the vicinity of a flame. Aseptic technique is required for the maintenance of pure cultures and the successful isolation of specific types of microorganisms. In the present day, laminar airflow (LAF) devices are used for all microbiological works, which are stationed in clean rooms or sterile rooms. Pure culture. A pure culture is a culture that contains only one species of bacteria, as depicted in figure 2. Mixed culture. A mixed culture encompasses more than one species, as depicted in figure 3. When isolating bacteria from the environment, the microbiologist always starts with a mixed culture. The samples are diluted and plated in any one of the techniques listed below to obtain a mixed culture and also to count the total population in the given sample. A pure culture can be obtained from the mixed culture by subculturing and streaking for isolation. Let's learn on the usage of the loop or the stab inoculation. Two different types of inoculators can be used depending on the purpose of the work, that is, a loop and a needle. The loop is used to transfer cultures from one medium to another and also to prepare bacterial smears, and lastly to streak the plates. The loop is the tool of choice for working with a liquid inoculum culture. The stab is used to prepare stab cultures and to pick single colonies from a plate. Stab is usually carried out with inoculation needles, as depicted in figure 4. Inoculation of an agar slant. Rest the inoculum gently at the lower end of the slant and withdraw it slowly upwards, moving it from the side to side. The surface of the agar should not be broken. This should leave a streak on the surface of the slant as shown in figure 5. Inoculation of an agar slab. Using aseptic technique, pick a single well isolated colony with a sterile inoculating stab needle and stab the needle several times through the agar to the bottom of the vial or tube, as depicted in figure 6. Replace and tighten the cap. Make sure the tube and the cap are well labeled. Let's learn on inoculation of an agar plate. In case of agar plates, there is a greater surface area of sterile media that can be exposed to contaminations in the atmosphere, as shown in figure 7. The key is to keep as much of the lid over or covered the open agar plate as much as possible. Incubate the plates at appropriate temperature and in a secluded container. This is normally carried out in an incubator. Never open the lid on the lab bench when in an open contaminating environment. Let's learn on the isolation techniques. To study the microorganisms in food samples, it is necessary to isolate them as mixed population and further from mixed into individual species. For isolation and cultivation of these microorganisms, basic laboratory apparatus are required. First of all, samples are taken from the food source which needs to be tested. These samples, if are not going to be analyzed immediately, are then stored at preferably 0 to 4 degrees Celsius in sterile containers. The first technique is serial dilution. For analysis, samples are then diluted by serial dilution method to get the microbial count. Serial dilution involves taking a sample and diluting it through a series of sterile diluent, for example, distilled water or 0.9% saline. Then a small measured volume of each dilution is used to make a series or pour or spread. By diluting the sample, it is possible to obtain an incubated plate with an easily countable number of colonies, that is between 30 to 100, and then calculate the number of microbes present in the sample. Procedure: take a sterile pipette, draw up 1 ml of well mixed sample or culture into the pipette, add the sample to the first tube. The total volume of this tube should now be 10 ml. This provides an initial dilution of 10 raised to power 1. Mix the dilution thoroughly by emptying and filling the pipette several times. Take a new pipette and draw 1 ml sample of 10 raised to power minus 1 dilution and place it in the second tube, which already contains 9 ml of diluent. Mix well as before. This gives a 10 raised to power minus 2 dilution. Repeat this for the remaining tubes, removing 1 ml from the previous dilution and adding it to the next 9 ml of diluent. Pour plate method. A pour plate method is one in which a small amount of inoculum from broth culture is added by pipette to the center of a petri dish. Molten cooled agar medium in a test tube or a bottle is then poured into the petri dish containing the inoculum. The dish is gently rotated clockwise for 3 times and anti-clockwise once. This ensures that the culture and the medium are thoroughly mixed and medium covers the plate evenly. Pour plates allows microorganisms to grow both on the surface and within the medium. Plating and counting procedure. Use a known volume of each dilution in plating. By starting with the highest dilution, for statistical purposes, replicate the plates should be prepared. After incubation, the plates will show a range of numbers of colonies. Choose the plate that has an easily countable number, about 30 to 300, and carefully count every colony. Using a marker pen helps to avoid counting the same colony twice. Then calculate the number of microorganisms in the sample by using the formula. Number of microbes per ml is equal to number of colonies multiplied by dilution of sample. Let's learn on isolation of pure culture in media. Streak plate method. A sterile inoculation loop is used for preparing a streak plate as shown in figure 5. This involves the progressive dilution of an inoculum of bacteria or yeast over the surface of solidified agar medium in a petri dish. The progress of streaking should be in such a way that colonies grow well separated from each other. The aim of the procedure is to obtain single isolated pure colonies. A culture developed from single unaltered colony is called as pure culture. A loop is flamed and then touched to the first area to retrieve a sample of bacteria. This sample is then streaked several times in the second area of the medium. The loop is then reflamed, touched to the second area and streaked once again in the third area. The process can be repeated in a fourth and fifth area if desired. During incubation, the bacteria will multiply rapidly and form colonies. Single cell isolation technique. In this procedure, cells from a single isolated colony is again subjected to serial dilution and plated. Colonies arising from such plate are considered as a colony from a single cell. Microscopic isolation of single cell. The serially diluted sample from the above procedure is taken in a hemocytometer and a single cell is aspirated using a pipette. Conclusion. This chapter dealt with the sampling of food samples for microbial analysis, its enumeration and methods for isolation of pure culture for further studies. These methods are common for the isolation of beneficial organisms and spoilage organisms. The methods have been standardized over a period of time and are consistent. It depends on the skill of the microbiologist to isolate the right organism. The sampling and its preservation is one of the most important steps to isolate the organism of choice, as it can lead to wrong results. However, if all the procedures are followed meticulously, it is easy to isolate and preserve an organism. Thank you, viewers.